My name is Daniel and I am in 8th grade. The most exciting thing about my project from is that I got to add to a real body of scientific knowledge. This summer I learned that researchers are making a mutant library of Volvox carteri. Volvox carteri are a type of green algae in the shape of a sphere. They are a model organism for studying algae. A mutant library is made by creating random mutations in wild type or normal organisms in hopes of seeing interesting characteristics in the mutants that will help researchers discover more about how genes in the organism function. We can also possibly find mutations that are beneficial to the organism or to society. I decided to volunteer to help with researchers in 3 ways. 1. To test methods of mutation to determine which UV intensity creates the largest variety of mutants in order to improve the speed and efficiency at which the mutant library can be made. 2. To screen mutants I create for new varieties and interesting characteristics. And 3. To look for ways in which mutants can be screened for possible increases in lipid production to improve biofuel researchers. Volvox have lipids, which are fats and oils that can be turned into biofuel. The reason people use algae for biofuel is because it can produce between 2,000 and 5,000 gallons per acre, which is way more than renewable feedstock can produce per acre. Algae does not need large amounts of cropland to grow, so it does not have to compete with corn and other crops. It also grows much faster than crops. The first step was to obtain and grow Volvox cultures. Cultures were grown in a growth chamber under full spectrum light for 16 hours and 8 hours of dark. 4 to 5 Volvox colonies were passaged into fresh Volvox media every 4 days. I spent the first month of my project practicing my sterile culturing techniques, making sure my growth conditions were suitable, and observing the behaviors of wild-type Volvox algae. This would help me distinguish mutant algae from wild type when I was ready. The next part of my procedure was the actual mutant process. In this part, I subjected mother spheroids to different UV intensities using a UV crosslinker. Immediately after UV exposure, the spheroids were transferred to fresh culture tubes. The tubes were wrapped in foil and incubated in the dark for 24 hours to keep the Volvox from using light to repair. I tested each UV intensity separately. The final section of my procedure consisted of counting and screening mutants. This was the most time-consuming section, and I spent about 3 weeks per UV test intensity on this part. Every couple of days I would rescreen the mutant cultures for possible mutations, isolate the mutants, photograph them, and continue to grow them to determine if the mutant phenotype was retained in future generations. My results were very good. When testing 5 different intensities, I found that the largest variety of mutants were created with 20,000 and 40,000 microjoules. This information is very important because it will allow researchers to expand the mutant library at a faster rate, therefore gaining more knowledge about gene function and Volvox more quickly. The next thing I plan to try is a mutant already mutated Volvox at these intensities to see if even greater variety of mutants can be achieved. When screening my mutants, I found a particularly interesting mutant that had not been seen before. I called it the double mutant, but I plan to pick a more interesting name later date. The characteristics of this mutant appear to be delayed hatching, with daughter cells, uneven sizes and shapes. You can see it here next to the blue arrow. This mutant has been isolated for further study. Here are a few more other isolated mutants without gonidia, which obviously couldn't reproduce. 1 kidney bean, these are dented mutants that are actually quite common, and 1 tiny culture, which we have isolated for further study. Many of the mutants screened express characteristics similar to those already found, like Reg A. In this slide, but this is actually still quite beneficial because not all mutants are passed on to the next generation. So researchers who study need to be given additional supplies of those mutant lines in order to continue their work. Some of my Reg A cultures were passaged on to researchers at Danforth Plant and Science Center. In my research, I was also able to document which type of characteristics showed up often under the UV intensities. This will be very beneficial to researchers who want to work with certain types of known mutants and need to continuously create stock cultures of them. Finally, as I looked through my mutants, I tried to pick out a few that might be further studied for characteristics that would benefit biofuel production with Volvox carteri. This is very important because biofuels can help decrease greenhouse gases. Reg A mutants appear to have more somatic cells, which is where lipids are stored. These are good mutants to study for biofuel. To continue this project, I would like to develop a quick screening test for lipid content in Volvox carteri, possibly using a Nile red method to stain lipids to find if the mutants that would be useful in biofuel research.